Hey guys, this is a video lecture to cover the rest of the material that we didn't get to cover in the last bank of notes. We ended on Thursday with this skin aging slide, and even this is a small kind of aside on the way through our investigation into Alzheimer's disease and brain aging. But skin aging is something that people do have to deal with as well. You can see clearly in this picture, this is a fellow that is a truck driver who's been constantly exposed to increased UV rays on his left side from driving a truck, and then not on his right side, and you can see the clear effects that has on his skin aging and skin function. There's general agreement that that's the main reason that skin ages, that it's UV exposure that directly damages DNA inside the epithelial cells. DNA is damaged, and also a protein called retinol, which is involved in maintaining the structural integrity of the skin. And because of the nature of the damage, that there's direct damage inside the cell, on average, topical creams don't seem to correct the problem. And if anything, they just sort of glaze it over or make wrinkles look smoother, try to blend different colors and spots together. So they don't actually correct the problem. And we're only just in recent years getting an idea of how we could correct skin aging. And it's really quite fascinating. They use lessons they've learned from octopus eyes, because the octopus sitting on the ocean floor, always looking up, always has constant exposure to UV radiation. Now, granted, it's less UV radiation than we would get on the surface, but their eyes don't seem to deteriorate even with this lowered exposure to UV rays. So they've developed some mechanism to protect that very sensitive tissue from UV rays. And if we can learn how they did that, that's the first step in being able to develop a way to treat aging and help protect tissues in humans as well. So it's really cool that something completely foreign to this topic, this tissue, seems to be able to provide us an answer into how we could further protect ourselves. Really cool area of research. Now, what I do want to spend more time on, though, is this idea of brain function with aging. And we had a lot of groups yesterday talking about brain function in the showcase, and a lot of really good work done on this topic. In general, dementia is a broad umbrella term that literally translates to without mind. It has Greek roots and translates to without mind. It's sort of the metabolic syndrome of brain function. So there's a lot of different characteristics that people can measure, and if there's 2 or 3 that are abnormal out of the whole bunch, then someone is generally characterized as having dementia. Alzheimer's, on the other hand, is a subset of diseases within dementia. It's a progressive disease that typically only affects memory, thinking, focus and attention, and it really affects newly learned information, which is why older people with Alzheimer's disease have trouble remembering things that have happened in the recent past, and maybe don't have as much trouble remembering things that happened to them a long time ago. Typically, 65 years is what we consider normal onset, although it's variable for everybody. And then Alzheimer's that develops before 65 years is termed early onset, only because we really need to find some general description to separate these categories out. And as we'll see, they seem to stem from different mechanisms as well. It's difficult really to study it, because we can't study the brain as invasively as we would like to, and we don't even really know what to look for. When we talk about things that go wrong with Alzheimer's disease or dementia, it's a list like this: cognitive function, memory, attention, language, problem solving, executive function. These are words that we use to describe the level in kind of the function of the brain where problems happen, but these can be connected. They can sometimes appear without other ones, so it's not an exhaustive list at all. It's really difficult to try to assess these because we don't know what it means to have a memory. How does that look when you have a brain scan? How do you test a memory that is damaged versus one that isn't? So really the question is, how do we study brain function? What do we compare before and after to see if there's been a change in brain function, either with age or with supplementation? And we use, right now, a lot of games, a lot of things that test the ability of people to accomplish a certain task. And generally those tasks involve the process of memory, involve the process of attention. So we don't have to know exactly how to stimulate it, we just make them perform a task that requires them to do that kind of activity with their mind. So this is actually a screenshot from a test that I used during my PhD research when I investigated cognitive function. And it's called the IMPACT test. It stands for Immediate Post-Exercise Assessment of Cognitive testing? Oh no, it's concussion testing. It's used in hockey players or any athlete to assess cognitive function before and after an injury to see if they can be brought back to play. So in this particular test, you're presented with a list of different symbols and you have to press a number corresponding to that symbol, and then the symbol at the bottom will change and you press a different number. It seems pretty easy, except that halfway through the test all of the symbols disappear. And so you have to remember the order of those symbols. You still get that one symbol at the bottom and you still have to press the number that corresponded to that symbol, but you just can't see the symbol. Another part of this test was spatial memory. So you'll observe a list of different shapes, and these shapes are arrows and they're stylized arrows, and you have to remember whether or not the shape presented to you was in the original list that you had to memorize. You say yes or you say no, you're told if you're correct or not. They do the same thing with words. They have number association and pattern memorization. There's also a test that's very commonly used in the literature called a Stroop test, where in this particular example, this was an app that I used on my iPhone. You're presented with a word up top that is the name of a color, but the name of that color is written in a different colored text. So here, the word orange is written in red letters. So your subject would have to look at that word, and then in the list of colors at the bottom, find the word red. So we're not matching the word orange, we're matching the color of the text. And you can see red is in the bottom right-hand corner, and there's an added layer of difficulty because it's written in yellow text. So you can collect the correct answers, your score goes up, you get 3 strikes before the test ends, but overall it's a good way to test cognitive function and what we call executive function, being able to focus our attention and time and reactions appropriately. Now, function, the performance of those games generally seems to go down with Alzheimer's disease, and we don't know why, but it seems that Alzheimer's disease is associated with the formation of what are called beta amyloid plaques. And you can see that in the picture here. This is a beta amyloid plaque. All those individual green rods are beta amyloid proteins. They aggregate together, and for some reason they impair cognitive function. They make these plaques. There's also neurofibrillary tangles that we don't really know how they cause Alzheimer's disease. We just know that in the brains of people with Alzheimer's, these seem to be more present, and that's what we're focusing on now to try to elucidate that mechanism. Now, we get these beta amyloid proteins all the time. Even right now, when we're younger, we're constantly making these beta amyloid proteins. They're cut from what's called the amyloid precursor protein. And if you look way in the background, just above and to the left of center, there's a blue squiggly protein. That's the representation of an amyloid precursor protein. So we cut that up and we end up with these beta amyloid green cylinders. And normally we can process them or dispose of them. But for some reason, our ability to deal with beta amyloid protein goes down. It is impaired when we age, and then it accumulates in Alzheimer's disease. You can see here the process of cutting that beta amyloid protein from the amyloid precursor protein. There's just enzymes in the brain that act like scissors and cut those beta amyloid green rods from the APP. Generally, it's not heritable Alzheimer's disease. The beta amyloid proteins will just accumulate and seemingly at random in a lot of people. But the early onset form, which is why we had to categorize 65 as the threshold, early onset Alzheimer's does seem to be heritable. And the reason for that is there's a gene inside all of us called the PSEN gene that encodes one of those enzymes. So the presenilin protein is named because it occurs before you become senile, I suppose. And that protein is one of the enzymes that cuts APP. Now, the mutation in the gene doesn't change the enzyme function completely, it doesn't make it inactive, it just changes it very slightly. So when that enzyme attaches to the APP protein, it does so in such a way that the cutting mechanism is just moved to the side a little bit. So instead of making a green cylinder beta amyloid protein that is 40 amino acids long, it makes one that is 42 amino acids long. That's the only difference. But that small difference has a really big impact because those longer beta amyloid proteins are a lot harder to degrade and they're a lot more likely to accumulate. So just that one very small change in function of the cutting enzyme results in a massive change in the ability of the brains of these people with this heritable mutation to be able to deal with the beta amyloid proteins that accumulate. So this could be one way that we screen people in the future, to look and see if their individual genome has this mutation, and hopefully in that time we'll be able to come up with a way to correct that, but certainly you'll be able to make preparations and at least know that you might be predisposed to Alzheimer's disease. So how does cognition change with age? We're told that it goes down, and we can see that here. This is a 6-year longitudinal study in humans, and this is done in older adults that are over 65, so that are predisposed to Alzheimer's disease. What I want you to see is they've grouped people into tertiles, into thirds. So there are different age groups. Oh no, sorry, there aren't different age groups. There's different tertiles based on VO2, so low, sedentary, medium, and then higher VO2. And at the beginning of the study, all of the, down here at the bottom, the modified mini mental state examination scores were the same. Now, we haven't seen this test before. This is just a common test that people use to assess cognitive function. It's just like the test that I showed you before. And the important thing is that the scores are all the same, and this test wanted to see whether or not those scores changed over 6 years, and if they changed differently based on the VO2 of these older patients. Now, what I said earlier does hint at one flaw, one complication of the study. You'll notice that there are differences in age. The most fit people also happen to be the youngest, relatively the youngest. They're only 66, versus the least fit people that are 71. So that's a confounding factor that we might have to sort through as we interpret these results. It's not just fitness that's different in these people, it's also age. And if you look further through this table, you can see that household income is different, and there's differences in hypertension that could possibly be related to fitness as well, or even age. But we would have to correct for these in order to know that these are not the cause of any changes in the cognitive test. So what happens over 6 years? In general, there's a bigger decline in cognitive function with lower VO2 max. Kind of interesting, because we've seen that fitness is such a potent stimulus to help improve muscle function, and now we're seeing that at least baseline fitness seems to protect against decreases in cognitive function. We know that fitness does go down with age, so maybe it's a moot point that we're inevitably going to see a decline in cognitive function as we age, because VO2 will go down as we age. But it seems as though the decline is greatest when you have the lowest VO2. And you can see in these graphs at the bottom, these aren't labeled, but the black bar is the lowest VO2, the white bar is the middle VO2, and then the gray bar is the highest VO2. And these bars indicate the drop, the decrease in cognitive function. So a bigger bar is bad here. What we pointed out on the last slide, though, is important too. The lowest VO2 max is also the oldest, so it would make sense for those people to see the largest decline in function. Sure, everyone's over 65 years old, but remember, Alzheimer's is a progressive disease. It gets worse as you age, so if you are older, it should be more advanced. If you're older, then it should advance more as you get more older. So it makes sense that if you're older, you'd see a bigger decline in cognitive function as well. It's difficult to know whether it's related to VO2, and we can try to adjust for hypertension and for age, as is done in the right graph. These are the same results, just adjusted for those factors that were different at the start. And it looks as though VO2 max still retains that same effect, but you don't know for sure until you actually do that test. The other thing that we need to consider is that on that last page, the MMSE was identified to have a maximum 27 points. And here we're seeing a decline in the performance of this test at a maximum of 0.6 points. So what really is the significance of this change? Does it relate to cognitive function in a real-world situation, or is it just a really small artifact of this test? In other words, can we translate these findings to a whole body change that we would notice in somebody? I'm not quite sure. So what do we have that we can use to treat brain aging? We're still learning about the mechanisms, and so treatments will be fairly crude and fairly rudimentary. But there are a few out there. One recently that's come up is coconut oil. This idea that supplementing with coconut oil can actually reverse or delay the onset of Alzheimer's, and we'll see a video about that shortly. I want to talk about omega-3s because it seems like these do almost everything. They're great for diabetes and insulin resistance. We saw earlier in the course that they help with depression after pregnancy, and now it seems like they can improve cognitive function, or we're going to find out if they can. I want to look more at those brain games and see whether or not regularly participating with brain games can prevent decline. The one thing that we really need to keep in mind here, like we addressed in that MMSE test, is can we translate these findings? That's the biggest hurdle in the brain game world. Is it something that we can measure a change in daily life and daily problem solving? Lastly, we're going to revisit calorie restriction because it's recently come out, and it's fairly surprising that this might be a way to effectively delay the onset of Alzheimer's. So I'm going to include in the notes below this video a link to the CTV News story about coconut oil. go and watch that now. It's only 4 minutes long, and we'll pick up with the rest of the lecture when you get back. Okay, I'm assuming that you guys have watched the video. Now that you've seen that one woman's story about coconut oil, we really don't have a lot of other information to go on. There's not much research on coconut oil and Alzheimer's disease, and you could really get that from the video. There's only one study that's now starting to look into this, and this was reported in May 2013, so we don't have information about it yet. It's really driven by her qualitative observations, and I question how unbiased she is, especially when it's her husband that is involved in this, and she's noticing changes in walking and clock drawing. I question how rigorous that is. If we look at what coconut oil is and why it would affect Alzheimer's, the one main point from the video is that brain cells seem to be starving, and coconut oil is a way to give them energy. Now, coconut oil itself is mainly composed of 2 fatty acids, and these are saturated fatty acids and medium chain triglycerides, medium chain fatty acids. So they're not long chain fatty acids like we typically talk about, like palmitate. They're medium chain, and they're saturated too, which kind of goes against a lot of what we know, that saturated fats are bad and trans fats are bad. So the 2 main fats are lauric acid and myristic acid. If we look into the literature, it seems as though lauric acid promotes the formation of fatty acid globules inside the brain. Now these are linked to Alzheimer's disease, and of course linked is a keyword that usually means correlation, so we don't know if they cause Alzheimer's disease, but at least there's this mechanism that we've uncovered. Lauric acid seems to make fat accumulate in the brains of Alzheimer's patients. So it might be akin to the athlete's paradox, the insulin resistance and type 2 diabetes thing that we talked about last year, that overcrowding of fat inside the cell might disrupt normal signaling, which would be a bad thing. Myristic acid, it's also required for a protein that activates the presenilin genes. So this fatty acid, this myristic acid, which is part of coconut oil, activates another protein that turns on that longer, the longer beta amyloid enzyme. So myristic acid contributes to more of the 42 amino acid beta amyloid proteins, which might mean that they would accumulate inside the brains and maybe contribute to Alzheimer's. It seems likely, at least. So these 2 main fatty acids in coconut oil seem to have detrimental effects as far as Alzheimer's go. Now, we haven't gone so far as to link them to Alzheimer's. The mechanisms that we do see with these 2 types of fatty acids, in theory, would contribute to Alzheimer's, but that remains to be tested. Now, the idea that the brain is starved may or may not be true, and if that's the case, coconut oil might be able to help solve that problem because of its characteristic of containing medium-chain triglycerides. You guys remember that medium-chain triglycerides are special in the body because they don't need to be transported across membranes. So they don't need to be transported across plasma membranes or mitochondrial membranes. They just slide right through. So if somehow there is this starvation occurring, if there's not enough glucose getting into the neurons and other brain cells, then maybe medium-chain triglycerides could circumvent whatever the blockage is to provide energy, possibly. But this is a fairly controversial area, and I always like to stay cautious, and I hope that that's rubbed off on some of you guys, because a lot of people will take this one woman's observations and run with it and say that this is the be-all, end-all cure for Alzheimer's. And I found one quote that takes it to the extreme. This is, as I was searching for Alzheimer's, I stumbled across another news story that reported on the same video. And inside said, coconut oil also kills bacteria, making it a natural antibiotic without the negative side effects. Teeter, who is a professor at the University of Maryland, and she's being quoted here, so I don't know if she said this, but the article says that Teeter said, because of that natural antibiotic characteristic, it also helps defend against viruses like HIV and herpes. So think about how wild a claim that is, and how unlikely it is. I struggle to know how to deal with this personally. I would like to say absolutely not, but again, we don't really know, we don't have the data. I'm inclined to believe that it's not the case. But a lot of people would just take this and think, oh, I have AIDS, I can eat coconut oil and be fine.